Hi
Hello!
there.
Welcome to Microbiology Journal Club. We nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to aiding the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology, and I've mostly worked on bacteria, but I've also worked as a research integrity specialist, and I'm currently working as an editor for a scientific journal.
Every week, we meet to talk about microbiology, and today it's our news week where we're doing an overview of some of the coolest microbiology papers, a bunch of SARS-CoV-2 stuff that we've seen in this last week.
And at the end of every month, we try to focus on doing a deep dive. So next month, we're going to be looking at how magnets were used to study antivirals. But if you have any papers that catch your fancy or you want us to look at, please message us at Twitter and we'll try and put up for the next poll for our deep dive.
Yes, absolutely. You can follow along with any of the papers that we discuss or that get sent to us by looking at our Shards of Terror library linked in the doobly-doo below.
And we want to hear from you, so please use comments or tweet us at microtwjc. And boy, do we have a show for you. This week we'll be talking about Omicron and antibody neutralization, antibody neutralization, and Omicron, and of course, the neutralization of Omicron spike by antibodies. Yes, there's a whole bunch of data about the serum neutralization of Omicron, and we're going to be talking about it through the first part of the show. But not only that, there are other microbiology stories that have dropped this week, like new antibiotic candidates and bacteria with tiny organelles that we can target, and finally, marine bacteria that can use dust that can use dust for food, apparently.
Yes.
So let's get this show on the road. We've been quite exhausted with the Omicron at the moment, so the point where I realized like my show notes just devolved into nonsense at that point. So I had to make. So we're gonna. But yeah, there's been a lot of papers that have been published over the past week about the strain. So let's take a look at some of them.
So
I'm
much.
gonna start off with
Maybe just
yeah.
everyone, I'm sure if you're tuning in specifically for this section, you know about this new variant, Omicron. A whole bunch of different spike protein mutations were found on this variant, first identified in South Africa, but later found in all sorts of localities around the world because we're only as good as we can observe in a specific region. And once the signature, I think, was disseminated to everyone and people started looking for it, it was found that it was in many other places. There's been scant data about exactly what those mutations mean. Because you can study mutations in different contexts, but having them all together in a single protein makes a big difference. And having that hard data, I guess, to say what is the effect of these hasn't been present until now. until now.
Yeah, and even the data now is somewhat incomplete, so we still don't. So we're not going to see anything about how, say, severe this infection is for a while yet, because again, it was discovered less than 2 weeks ago,
Yep.
and the course of infection can be a couple of months, so we won't be able to hear anything about that until then.
Yeah.
Also,
I was gonna say also, it's not necessarily the dominant strain in the dominant variant in various localities. I think Delta is still a very large proportion of the infections that we're seeing. So just keep that in mind as well. That if you want it, in order to see that sort of the data about outcomes that's going to come from large swaths of people being infected. Well, hopefully we never see that data, right? I mean, infection levels are just kept down, but we will only see those answers from the sort of the quantitative side after the numbers come in, right? After those infections have happened.
Yeah, and it's also I think important to point out that it was discovered in South Africa. We don't know for certain where it actually came from,
Yeah.
because I think there have been some studies that have found it may have emerged in other places earlier.
Yeah.
But I'm going to be starting off by looking at the papers from South Africa, because that's where most of the data is from. So I think we'll go for the first paper, which is titled. Let's see if I can pull it up quickly.
So this SARS-CoV-2 Omicron has extensive but incomplete escape of Pfizer BNT elicited neutralization and requires ACE2 for infection.
Yeah, that's right. So this has got many authors who were instrumental in alerting us to this. So I want to highlight Tulio de Oliveira, who like when we first
Yeah.
reported on Omicron, I like used a bunch of slides from his talk in our discussion.
Yeah.
So he's quite like been quite important on that, and he's on a
Active
tear
on
on Twitter
Twitter.
about this.
Active on Twitter.
Very active on Twitter.
Uh-huh.
I highly recommend. Good follow. Um, so yeah, they look. So actually, one of the cool things they did. So just from a technical perspective, we see a lot of papers that use a viro like strain of cells. So that's from
Hmm.
green monkey. It's used to grow SARS-CoV-2, and in previous studies they found that if they grow it too long, it loses their cleavage site, their furin cleavage sites, because it's not.
Yeah.
It kind of it. But they've actually developed their own human lung cell line, H1299 ACE2, to do basically the same job that people have been using viro cells for. Someone give a shout out to that because that's quite a cool little new technology.
Yeah.
Um,
Yeah, so right, I
but
think
yeah,
just to clarify, like the when you passage SARS-CoV-2 in viro cells, they pick up mutations.
yes,
Yeah, like something, I guess
yes,
viro cells have like a different protease mix in them, and so yeah, it's not so important for yeah, the furin cleavage to happen in order for them to get inside.
yeah. So if you like grow SARS-CoV-2 in those cells over and over again, it stops looking like natural SARS-CoV-2. And whenever you analyze those passage cells, you have to take that into account that it will have some
Right.
mutations that you'd never see in real life that are
Hmm.
actually maladaptive.
Hmm.
But so anyway, let's get to the meat of this because this has got some juicy figures. So we've got like some
Yeah, so in this one,
looking.
they have serum from people
Hmm.
who've been vaccinated by the Pfizer vaccine,
Yep.
or vaccinated and later infected, or before infected, either or.
Yeah, and they test like the neutralization titers uh,
Hmm.
of say of Omicron versus the like D614G variant, and they seem to be the number I'd highlight here 41.4 like fold less. So I mean this is a logarithmic graph, so you might not think it looks like 41 fold, but it goes up in steps of 10.
Yeah.
So I think like someone on Twitter pointed out that 41 is also about the same amount that antibodies get boosted in a paper that I haven't cited. So I guess, yeah, that's not a fact I should really be saying on this because I haven't got my source. So I sourced someone on Twitter. I'm sorry. Ignore that.
But yeah, like this is in line with the prediction, right? There was the prediction, we saw mutations that it would lower the effectiveness of antibodies. Again, to remind people, antibodies are this what we've come to understand as one of a correlate of protection for SARS-CoV-2. It's how in the clinical trials before we had efficacy on severe disease, people were looking at antibody titers and evaluating our options based on how many neutralizing antibodies are made. But of course, neutralizing antibodies go down over time, but you're left with the
Hmm.
memory that's supposed to, revitalize those neutralizing antibodies at some point. But these particular assays that we're looking at are just that initial instance of like the collection of antibodies you have at some given moment. Is it preventing SARS-CoV-2 from binding to cells and getting inside?
Yeah, I guess we should probably go through like what these graphs mean because I've got a couple of these lined up. That
Right. Yeah,
the
it'll benefit for
PRNT50.
all future discussions of this data that we'll look at.
Yeah, so essentially, I think what they do is they try and find the concentration of antibodies that will neutralize 50, that will prevent 50% of cells in a certain like
Hmm.
model being infected. So in this case, they got their H1299 ACE2 cells, and they want to make sure that those don't get killed off. So they I think through a statistical thing, they try to look for the 50%
Yep.
rather than 100%. Um, and then they plot that on a graph. So this graph here it shows the threshold of neutralization for each person's serum. So these are serum is taken from people's blood, so it's a blood sample from like one time point,
Hmm.
and when you spin it down, that serum has a bunch of different things, but among them is antibodies.
Hmm.
And so the idea is that the antibodies in this serum, if you plug them into a petri dish with your cells and the virus, the antibodies will bind to the virus and stop the cells from dying. And if you up the higher the concentration of the serum, or the or actually what they actually do is dilute the serum down to the
Yep.
lowest amount first.
And
So
then yeah, and
they
they test along the whole range of dilution. And that's just to get the resolution, right? To find some important points.
Yeah, they want to because they don't nobody knows exactly how many antibodies they have in the serum. This is just a way to figure out how to ballpark that without trying to count the number of antibodies. Um, yeah, and so ideally, like the lower the more you dilute the serum, the less theoretically less potent the antibodies get. So if an antibody
Yeah.
can still neutralize 50% at really high dilution, that means that it's really strong. It's a really
Yes.
strong antibody response.
Yes.
And
Yes.
and in order to display it here, they actually they take an inversion. So actually, the higher numbers mean the neutralization, I believe. So here you can see with a D614G, that's those antibody
Yeah.
serum, those can neutralize that virus much easier.
That's right. That's right. And
Yeah.
I mean, another thing that we're seeing from this data, and we only covered it sort of slightly, but like having a previous infection does give you a suite of antibodies that can add some extra neutralization ability on top of it. And what we'll know, we've talked about it a little bit, but like those antibodies are different and not necessarily protective on their own that from vaccine antibodies. But again, this is just like a snapshot assay, but you see that effect pretty clearly between the teals and the golds. But each of those samples are paired with, because they were tested both in the wild type or the ancestral version, the D614G, and the Omicron scenario. And you can see in all the paired samples, the slope of that line is going down. So
Yeah.
there was the reduction in all of the paired samples.
Yeah, so like this person like has in their sample, you can see that line represents the difference, and that's just still one person. And the only time where you see them really cross over is this line, and this red line is kind of around the limit of detection, I think. So yeah, so we're going to be rushing through a couple more. So this next paper is also has one scientist who's based in South Africa, and this is a very preliminary report. I have to highlight this is even a preprint because this person shared it on their Google Drive on Twitter. So this is very early, as in for they're still working on this right now. So this shouldn't be regarded as a final idea at all.
Yep.
Yeah, and I
But
keep
they're
worrying
seeing,
because
oh, what are you worrying about?
yeah. Oh no, I was worrying because I worried because they left their Google Drive, and I'm concerned that I might accidentally write stuff on it. But yeah, they compare. So previous study they compared the wild type and the Omicron. This one they compared the wild type, the Delta, and the Omicron to see differences in neutralization data, and effectively this is showing a similar thing, where like Delta has slightly more neutralization, and Omicron is. They find here that it's about, and here they're doing something slightly different. So they're using a
Yep.
pseudovirus. Now let me see what because this is because
They essentially replicate
so
they
yeah
replicate all the mutations that they see in the Omicron spike on a spike that then gets packaged into not the into the envelope of a different virus.
yes. So yeah, what they did is they created so since that the structure of Spike has been released online, they could like print out a version of that and then put it into a different virus. In this case, use a lentivirus that can infect cells and produce a spike, but because it's a different type of virus, it doesn't infect humans, so there isn't a risk of creating like a dangerous variant. Um, and yeah, they looked at like 17 random recent blood donors in Stockholm and 17 previously infected hospital workers. So this is with blood donors and this is with hospital workers, and what they found was hospital workers seem to have a better response. Again, this is very early days, but might indicate what Daniel was talking about earlier. What if you had a previous infection, that might give you a slightly wider response to spike.
Yeah.
Yeah. So again, this is very short. They but and very early on. So I'm going to move on quickly to another preprint. This one is from Austria, I believe.
Austria.
So yeah, this is from a. Let's see. Let me double check where they got this. Oh yeah, this is from a variant that was isolated in Switzerland. So there's a variant of Omicron that was isolated in Switzerland. Um,
That's so
so
yeah,
yeah, this is
one
so
one five two nine is is pointing to like another is like a subcategory of Omicron.
yeah. So the thing that I was looking for is because like now we're away from the South African papers. One of the things I was wanting to know is like where are people actually getting their versions of Omicron
Mm,
from? Because uh, thing because this is actually a kind of an issue that's pretty because travel bans have really made it very hard to study Omicron
hmm
outside of South Africa. And also, it's made it quite hard to study Omicron in South Africa because there's constant delays on getting reagents and things like
hmm
that. So one of the things I was looking out for because I'm always kind of concerned about like uh like acknowledging people for their work.
Right.
And one of the complaints I've heard is that there's been a bunch of papers released where people have just taken a lot of the data that's been produced by South African researchers and republishing it almost as their own.
Mm,
And then so I mean this happens every time a variant comes out. I have to say every time the first paper that gets to into a preprint or like an archive is never from the people who actually initially discovered it. It never comes from the GIS aid researchers. It's usually someone who just downloaded that paper online, did a quick either did a quick addition to a paper they're already writing, or ran it right through a computer simulation, and you find that online. And I tend to skip over those papers, but some unless they have something, they add something significant to
hmm
it. Um, but that is an issue, and that's kind of a let me see if because apparently you're not allowed to swear when you're 5 minutes into a YouTube video, so I need to check what the timing. Yeah, no, that's a dick move. That's a jerk move if you do that.
I mean,
Oh, we're 17 minutes
yeah,
in, so it's
we're good.
fine.
We're good.
But
But that's like I mean this is just like the publishing environment, right? And like in terms of
yeah,
like the excitement around covering stuff from SARS-CoV-2, it just drives people to add the most recent, right? Like the most recent news item into like some of their current manuscripts in preparation. But then maybe it also means that it's hard to attribute like what group was maybe like most instrumental in bringing that data to the community, right? Something like this.
yeah, and it sucks for that group because they don't get the citations that they need to like to because that like help that changes funding, it
Mhm.
changes like your career aspects.
Right.
It is kind of unfortunate, and I feel bad when I see a paper that does bring something new but doesn't like give the credit where it's due.
Mm.
Um,
So how does that relate to
yeah,
this paper that we're talking about?
it
Oh,
doesn't.
okay.
This is my entirely different rant. that
Gotcha.
that this is an entirely unrelated rant to this paper. This paper because this paper but the rate related to this paper is and the papers that we're going to see is how they actually find their version of the Omicron to test
Gotcha.
with because I think people have to like find different ways to study this
hmm
variant because of this issue with the travel bans in South
Right.
Africa and
Yeah, so for
yeah
example, in the previous
yeah
paper, the
so
pseudotyped virus, that's a one way around it. You don't have the virus itself. Well, first of all, it might be hard to work with the virus itself because like that requires a certain biosafety level. So like making a
yeah
pseudotype virus is also just to transcend that barrier, the particular barrier of working with like a BSL-3 organism. Um, but in this one, how are they getting there? Uh,
they got it from from what I can tell they got it from like so they've got like the isolate number and they isolated so this isolate came from like Tyrol, Switzerland
Gotcha.
if I read it
Gotcha.
correctly.
Yeah.
Yeah. And they do so again this is another quite short paper. They do some assays looking at like the different vaccines and how so they get collect serum from people who have received different
hmm
vaccines and then they look at how those titers do against different
hmm
variants. Um, so they've got like the three main variants. So B.1.1.7, that's I think the alpha variant. B.1.3.5.1, that's a beta variant, and B.1.6.1.7.2, that's a delta
hmm
variant, and B.1.1.529, that's the Omicron variant. And so you can see like kind of how different vaccines and also people who've had a strain. So if people have recovered from the alpha, beta, and delta variant, and how those sera compare.
hmm hmm Yeah, and we're seeing that
So
reduction as well here.
yeah, and yeah, it's quite interesting, like how you see like the alpha. So in each one, you kind of the highest neutralizer is always the one that is infected. So
Hmm.
that's kind of a nice thing that I'm glad I'm seeing here. I think it's for the first time I've seen it, but yeah. Also, but this kind of builds up to the story that the serum antibody responses are not doing what we want them to do against Omicron.
Yeah.
That Omicron is antigenically quite different
Hmm.
from the
Hmm.
rest of them. Although, uh, I mean, some vaccine combinations do better. So it looks like
Yeah.
the
Chadox plus
BNT162b2.
plus Pfizer, the cross. I mean, these are those mysteries of the
Yeah,
immune system, right? Like, I think we've said this before. Like, vaccines give your body an immune response to something in a way that's like can be similar to what it might see in some natural version, but actually usually is trying to shortcut in some way. And that's like the magic of adjuvants and like why people are sort of excited, but also I guess confused about like what the benefits of the mRNA vaccine style is, besides like the rapid prototyping. And so yeah, getting a very it's almost like maybe it's like because they're being exposed, right? Like the second factor, the second signal to the immune system there is different in those two circumstances. I don't know. That's like one off the cuff
oh,
thought that I could
that
throw.
there's another thought I had is that also the timing of when these were given, because Chadox actually
Ah,
came online quite a lot earlier than
yes.
BioNTech. So what I'm
A
thinking
bigger
is like
gap in
that if
between.
we're seeing a yeah, if we're seeing a vaccine waning effect, then the Omicron can build off of
Hmm.
that. So whereas like things like so BioNTech is a lot more commonly used now, so things like the Chadox BNT162 could indicate that they've been that this is a recent change because I think they
Yeah.
only allowed like cross vaccination quite recently,
Yeah.
so that
Actually, that
could be
the
another factor.
data kind of like supports your thought there, right? Because the the Chadox neutralization for just B.1.1.7 is lower. Sorry, the double vaccinated Chadox, right, is lower for yeah.
Yeah, yeah. I mean, just looking at looking at the baseline levels, yeah. I mean,
Hmm.
looks like that is that is having that
Right.
effect across
Right.
everything.
Right.
So
Okay.
yeah,
Uh,
so that's a cool.
and then we have reduced
Yeah,
neutralization of SARS-CoV-2 Omicron variant by post-immunization serum.
yes, this is from the UK, from a variant that was discovered in the UK community, and they also did some tests using some. Uh, I think they looked at serum from people who had been vaccinated with different
Hmm.
things. Let's see.
Yeah.
Let's
They
pull
also
down
have
to the graphs.
Chadox versus Pfizer.
Yeah, so they've got. So this has got.
The
So
resolution on these
again,
graphs are
let me
really
zoom
rough.
in.
It's really
Oh wow, yeah, okay. So Chadox and BioNTech. Uh, yeah, someone used the bold font too much, I guess,
yeah, or something.
or used the wrong like.
Yeah, or maybe it was
But yeah,
exported like not at a high quality image.
yeah.
Because it was
I
made in
mean,
R. It looks like an R
sometimes,
graph.
yeah. Oh god, yeah.
Right. So it's like,
You
yeah,
flashbacks to working with R.
yes.
Uh,
Looks
but yeah,
good.
as I
Looks good in the
refer to it as
dashboard
R.
when you're playing around with it in the program, and then now you have to like send it out into the world to other people, and you have all these other concerns that like you're not thinking about as a scientist sometimes. Like, just like is the DPI right on my export setting?
Yeah. I mean, there. So again, we've got like lines that are linking that are showing like
The
each
paired
individual person's
samples,
responses
yeah.
to.
Or I guess not
Yeah.
paired in
So
this
you've
case
got
because they're testing. I don't even know what the first condition is. I assume that's
Vic,
wild.
Victoria. I think is the name for
Or
the original original sample
gotcha.
before even the alpha variant.
The native.
Beta is the old version of the South African variant. Delta is the one that's everywhere, and we've got Omicron, which is really low.
Right.
Um, so yeah, for Chadox, it doesn't look like there's been that much of a response. But again, I think that my thoughts on like the timing and the waning of vaccines, I think are
Yeah,
also an
consistent.
important thing
Yeah.
to take into account.
Hmm.
But I'm also saying that because I received the Chadox vaccine, so I've got a bias there. I want it to have worked, but
But so like I guess, but the point like always to make again is that this is this is what was predicted from seeing the mutations. Now we're seeing the data from it, right? To
Yeah.
say that the antibodies aren't as effective, but this is just a snapshot, right? This is a biochemical test in some ways, right? With some biological output. It's saying that the antibodies that are circulating at the moment of collection, right? At the moment of collection of that blood sample, those antibodies aren't doing what they should be doing to the virus. So the idea is that if the virus
Yeah.
did get into you, right, that first virus that first inoculation that you might get will encounter fewer antibodies, right? And so that virus could then maybe infect some cells. However, if those virus infected some cells, then your immune system would turn on and it would start making more antibodies, right, from its memory. And so like that's one thing that we're not seeing in this these snapshot
Yeah.
pictures is that yes, this is a correlate because we know we just don't want our cells to get infected by that virus. It would be great if every virus that came in just like never found its home inside one of our cells. But in the case that that doesn't happen, you still have a memory response, right? That would then make antibodies
Yeah.
and suppress that infection. So that could, and I think, again, this is maybe sort of trying to broaden the discussion to what we think would happen in the sense of like a vaccinated person who gets Omicron, right? That person still might be protected from some rapid expansion, right, of the virus in their lungs or their blood because they would get that memory response kick in.
Yeah.
But maybe that still means you're infected in some time, right? Like you still might end up transmitting it to people. You still might feel some mild symptoms, right? We don't know, like who knows about that scenario, but but that's how it relates to the data that we're seeing in front of us.
Yeah, none of the papers we've seen so far have looked intensely at like T cells or B memory cells, and it's likely those are still there.
Yeah,
But again,
doing something.
this is just like that first like 5 minutes of being of your SARS-CoV-2 being in your body and
Yes.
not being attacked immediately by antibodies.
Yes. Yes.
Um,
Yes. Yes. Which again,
so yeah,
it's
we've
a
got
concern because we would like to just not have it spread through our population, right? That's the goal, I think, in terms of like controlling the pandemic globally. But again, like from the personal protection point of view, right? Like it's also you have to take that the danger. It's not like a clear danger sign necessarily. Yeah, personally.
Yeah, this is a paper from Germany with this is obtaining from isolates that were obtained from people who'd returned from so let's see they'd actually come from it. So this Omicron strain was gotten from someone who had returned from Zimbabwe, and they were double mRNA vaccines, so the Moderna. Um, and they look at the neutralization based off this isolate. So again, we've got very similar graphs. Again, looking at this time, there's comparing the different vaccines, and I think in gray it's the Delta variant versus just the Omicron variant. And yeah, we're seeing like neutralization numbers. Again, these are just the antibodies, so we're not trying to cause a panic. But and also this is including boosters as well. So some of these people have been boosted. Um, So we see that the people who've been boosted tend to have tend to do a lot better if they've been boosted quite recently. So I think they separate out. So if you've been boosted within the last like 3 months or something like that, it does a lot better. And
Yeah.
this again shows that BioNTech does a lot better as well.
And that actually speaks to what you were saying before, right? That like our circulating antibody levels do go down over time, right? And a booster essentially resets that up to like some fresh level.
Yeah, I think that basically like because even though like Omicron is quite different from the other other variants, having enough antibody can negate that somewhat. I think what we're seeing like that if you have enough antibody, they can overcome that difference.
Yeah.
Um, yeah, okay, and this is the final one. This one actually got published, so that's something. So this has been through peer review. Let's see what the figures say, because again, this is just one figure, and oh, okay. And this is again comparing the different variants with. So this time they're using the control as like the baseline. So the baseline is the D614G variant, and then they're comparing the neutralization across all of them. And Omicron tends to be like the. They also test Mu. I've never heard of Mu before. I don't think we've ever talked about it, but
Yeah.
apparently that's also very immune evasive. And the thing that really stuck out was this. Come on. Uh, I Okay, right. I'm going to pull up the PDF because the sometimes publishers' websites aren't great, but that's fine.
Not everyone can do, e-life level.
Yeah, not everyone can do that. Or post just a PDF. Yeah. So here, like this heat map kind of tells quite an interesting story for me. Because so at the top line, that's just each individual patient who's been given a number instead of the name to predict their identity, and then on the y-axis is the different strains, and then like the heat map is like the hotter it is, the more the antibodies respond to it. And you can see that some of them that some people have different varying responses to antibodies. So you can see like there are two people who have very low responses just across the board, but Omicron just has this big blue line that just looks like it's hard to neutralize across the board. So, but again, this only tells part of the story.
Yeah.
So
like this is like one element of the variant in terms of when it first hits somebody's body, right? That what's circulating in the blood at that moment might not be able to control it at that moment. So like it will go on to infect a few cells, but then that infection will, if you're vaccinated or previously infected, it will trigger some sort of immune response. And then that part of the story, we don't have as much data for, right? Then we're back into speculating on, well, then what does it mean to have certain titers? How does that correlate to different disease? Are you susceptible to, right, severe disease or not, right? All of those questions are then coming to bear.
Yeah. And also because I forgot to mention that this was collected from an isolate discovered in Hong Kong. So again,
I think that was in one of the early reports, right? It said Hong Kong was also in the first surveillance event.
Oh yeah, I think this is one of the first places it was discovered. So again, we're getting like different like people having to like wait for the Omicron to come to them before they can do work on it, which is not amazing. So yeah.
Minimal actually left us a little comment. He can't find the link for it, but saying that like the lineage, right, like the Omicron lineage may also have some variants inside of it as well, right? I think that's definitely to be expected. And I think one of the things that I was interested in when we first saw the phylogeny of Omicron against everything else is, are there also like variants that would show like how it's related, right? Is it from stepwise evolution from some of the other more established variants that we've seen? Because everyone said like, oh, it's this all this cluster of different mutations. I'm like, how did those happen, right? Did they accumulate over time? Did they like recombine in from a specific other lineage? Like those questions weren't really answered, and it's kind of crazy because well, first of all, like it's very impressive that we have this type of resolution, first of all, to even like begin to even ask these questions or think these questions about like the viral evolution across the globe. But of course, like that surveillance isn't perfect, right? Like there's not a sequencing center in every locality, and this virus is spreading everywhere. So like sort of, I think seeing Omicron pop up like that also made me just maybe like a reality check on like that. Yeah, it's great that we can see this in some places, but there's also stuff happening that we won't be able to see, right? Mutations and lineages that we aren't seeing and not talking about because they're in places where there is no infrastructure to begin to even ask these questions.
Yeah, I think when we first talked about it, we got some like brief early phylogeny where they like had this big branching tree graph, and Omicron was just so far away from everywhere else, and the intermediate steps between the other strains and Omicron are really massive, so it's hard to figure out where that was rooted.
Right.
And there's been speculation that it came from an animal host. I think there's been also speculation it came from a convalescent patient, because I think
Hmm.
we've been looking at a couple of studies over the past year or so on people who've had convalescent had long-term infection with SARS-CoV-2, and those strains end up having quite a lot of mutations, as that has a much longer fight with the immune system, so it ends up
Right.
evolving in that situation. So there's been a lot of speculation that it could have been a cluster of immunocompromised people, or there's
So
some kind
there's
of
some sort of like,
index.
yeah, in intra-host like speciation, or like intra-host evolution, and then escape into the wider population.
Yeah, I don't think we're gonna get so we may never get solid answers actually,
We
but
never
I
get
didn't.
the answer,
But
yeah. It looks like a minimalist
yeah,
poking that there's some people, I assume these are scientists that he's listening to on YouTube, talking about the likelihood of recombination. Yeah, that's interesting. I mean, we have
oh
to
yeah,
look that up.
I saw this on Twitter as well. There was a fragment of a human gene found in a version of strain, I think, and
Hmm.
that's why people. That's one of the lines of evidence for it. It was evolving in people, and then it might have captured a fragment of a human gene.
Oh, interesting. I'd be interested to see like when that gets formalized into a document. We can dive into
Yeah,
it a little bit. Yeah.
yeah, because I find it very hard to cite researchers and publish on Twitter, because even saying that makes me feel slightly ill.
Yeah.
Um, but
Okay.
yeah,
And that brings us to the last paper I think we have in the onslaught of Omicron information.
yeah. And I hate to disappoint you. This one isn't about doesn't involve serum. This is about well, this does involve a little bit, but it's mostly about surveillance. So in the
Hmm.
UK, we've got a, a surveillance strategy where I think people are regularly tested in the community,
Hmm.
and from those tests they found a couple. So they found I think 581 symptomatic Omicron cases, and this is out of 56,000 other cases. So Delta is still dominant at this time point. Um, and so yeah, Omicron has a foothold, and they found that this. I think they found based on S gene dropout. So that's when you do a test for a couple of genes from SARS-CoV-2 and find that spike doesn't appear, and that happens because of a small deletion in the spike gene that Omicron happens to have. That is I can't remember what it does to the immune system, but has made it very easy to track, which
Hmm.
is good. And also, yeah. And then, so based on this data, they look at all these 581 patients, and then look at their vaccine records as well to see whether they've received the Chadox, or whether they've received the BioNTech, and whether they've received a booster as well. Uh, and and they pull out some data from that. And again, this is observational data, so there are quite a
Hmm.
lot of kind of confounding factors that are
It's
here at the
observational
moment.
data,
So
but I'll also say it's for the endpoint is also more clinically relevant, if you will, right? Because it's looking at
yeah,
actual case, like PCR positive cases, symptomatic cases.
yeah, and again, this doesn't actually say a thing about the severity. This is people who've come up positive, so
Yeah.
I don't know how ill they were. They just happened to
Hmm.
have the virus.
Yeah.
But yeah, so I think they look at how so people who've had the booster dose are doing very well in terms
Hmm.
of vaccine effectiveness uh,
Hmm.
with Omicron.
Hmm.
Um, and this is with Chadox with BNTs. So but if you're like me, and so I think Omicron is in grey and Delta is in black. So
Hmm.
Delta, you see, there's some vaccine waning effect over time. I think let me double check whether I've got this. I understand this right. Because I think that's the number of days since dose two. Uh, uh yeah, maybe. So I don't understand why that's going up, but
Sorry, what?
um,
Why
but
is it going?
oh no. So yeah,
This
so
is
I think
like
it's
how many days? Yeah, it's how many
time
weeks since
how many
since
weeks
the
since
dose? Yeah.
vaccine?
Hmm.
Yeah.
Yeah,
Um.
so it's going down, right? Like this is the trend of like circulating antibodies, right, going down or whatever.
Right. No. Yeah. No. Yeah. I understand that now. I think.
Oh, and then
Um.
why is it going
But
up in Omicron?
that.
Um,
Yeah. I
maybe only
think the main ex.
because. Oh, yeah, I'm not sure.
I, well, I think the sentence they put in here is that the the data from AstraZeneca might be really unreliable based on basically relatively small numbers, and they're more likely to reflect an older population. And
Hmm.
so these are like subgroup analyses within subgroup analyses as well.
Yeah.
So they're they're trying to sometimes there can be weird things that pop up, but yeah, it seems like
But I
BioNTech
think
seems
that,
to yeah.
yeah, I think the
Sorry.
takeaway though is that booster is really, it does provide this jump in vaccine effectiveness, right? So like, to me, what I take away from this is like we've seen like how the the lowering antibody titers, yeah, the lowering antibody titers, they're not as good against Omicron.
Mm,
Um, for neutralization abilities. And then here in vaccine effectiveness, which is a different endpoint that's looking
mm,
at like PCR positivity, the booster, which we know would increase those antibody levels, is giving us also some protection against spreading the virus around. Um, so I think
yeah.
it's to me it's like a bit of a justification for rolling out booster shots. Yeah.
Yeah, and like, and all I can say is that I feel like I chose really wrong with Chadox now. Oops.
No,
Um,
but like that's just
but
you get the vaccine that's available to you, right? Like it's not really
yeah,
I feel like not everyone's gonna have like a choice in that instance. So it's
true.
more
I mean,
yeah, it's just taking the tools that are around you and using them to the best of your abilities.
yeah. But I think I should also point out this is again in the realm of pre-preprint, so it's still
Hmm.
very early days. This is almost the first release of data, but they're still collecting it at the moment because again, this is they're capturing a snapshot shot that's very early on in the outbreak.
Yeah.
So there's a lot of things that, and I think they in this paper, I think they do take some like pains to try and say that this data isn't quite reliable at this point. There's the confidence intervals are very high, which
Hmm.
means that basically like a lot of this data could be due to random variation, which means that um, it might completely change.
Hmm.
Um, so, but I
So yeah.
thought that this was kind of important to show because uh, it's it is different from antibody neutralization,
Yeah, no, I
and
think
I
it's
think
really great to show it because of that. Like it's really like
yeah,
it's easy to get like to go down like a railroad track of like you want to find like the one assay that's going to give you the answers that you need. But like the reality is that like every assay sort of shows you something slightly different about what the effectiveness of certain interventions are. And yeah, it's
mm,
nice to see one where they aren't just looking at like the biochemical outcome.
yeah. And I think like this, I have to point out we are have to make decisions on imperfect information because if we wait for perfect information, it might be too late. And
Right.
so another
Right.
reason why I guess I showed this is because this is the paper that kind of made me book my booster dose appointment because I
Mm.
really didn't feel like it was that important until I saw this data point.
Yeah,
So
because I get
yeah,
this
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 is
this one
yeah,
might be one that someone cares about more. At least for me, right? I would be like, yeah, because I don't want to like pick it up accidentally. I'm not gonna get sick, but then I send it off, right? Like
yeah.
then I go and see some family and I send it off into the family. Like that's yeah.
Yeah, it's not perfect information, but at this point in time, I feel like that's enough for me to tell that because the cost for me, I'm so lucky to live in a country where I don't have to pay for vaccines, so
Right.
the cost to me is negligible.
Right.
Um, so
Yeah, and I just want to follow up. Like, yeah, I'm not. I definitely don't want to say that I'm endorsing some recombination theory on like how the variant came to be. It sounds like there's already a robust discussion, perhaps on Twitter and other sources. So I encourage people to look at that. But it is one of those things like, yeah, we may never have the answer as well. But it's fun to like with all this data, I think like from the microbiologist in me, like is always very excited in all these discussions about SARS-CoV-2 because there's so much attention on it. So like we have this amazing resolution and interest in actually getting data, and then we can see how the story is evolving, how this biological object, this virus, is changing in our world over time. It's just exciting to speculate. And I suspect that is also that drives some of the news and stuff that gets covered, right? Like there's just like, yeah, the details are there. Maybe it doesn't really impact your daily life because daily life is like make sure you're wearing your mask in crowded spaces, make sure you're getting your
Mm.
vaccine, right? Make sure you're doing those things haven't changed. But like
Yeah.
uh, trying to figure out what's going on and like all the different lines of evidence that could help you get to some of those answers. Like that's why we're here talking on the internet
Yeah.
about it.
I mean, I'm now not going to be watching Spider-Man No Way Home in the cinemas, so I'm upset. But Omicron. Um, but yeah,
But
I think
yeah.
that brings us to the end of our Omicron discussion.
Hmm.
Uh,
Oh
and
yeah.
so,
But then we do
yeah,
have one more SARS-CoV-2
we
paper.
do have one more SARS-CoV-2 paper, and it's a bonkers one. I'll be honest.
It
And
is
so,
bonkers.
if you're not ready for craziness, then feel free to tune off because we're about to skirt the line of pseudoscience here. I feel, but
Yeah, so
so
in this paper, they they have uh, the fecal samples from people that had positive RNA RT-PCR for SARS-CoV-2, but then they continue to culture those fecal samples in like a nutrient medium that's designed to cultivate fecal microbiota, and they continue to test for SARS-CoV-2 RNA, and they find that that goes up over time. And so,
yeah,
yeah, you want to say
so
some
yeah.
mitigating factors
Uh,
there before I go on?
oh no, I think we don't usually to go into the to tearing apart papers on here, so I'm kind of nervous about doing this, but but yeah, no, I have issues with this one. So like for a start, so I think they just use the RNA load to check for things, right?
Yes, yes, yes.
Yeah, they. So the thing about like some of those like using RNA is that sometimes if you like run those experiments for long enough, it'll those primers will bind off of anything.
Hmm.
So it's
Hmm.
this thing that happens with PCR, and I think it happens with where you will get a signal if you just run your experiment long enough.
Yeah, so
Also,
they're using
yeah,
Luminex. I'm not sure if they do an amplification. I think they do. At least at
they
the beginning
use
they do, but then they move over to using like a capture paste, right? But they could still do it. It could still be amplified. That's for sure.
So yeah, they use NXT tag sequencing for detecting three virus gene targets. So this is actually a kit that they got,
Hmm.
and so I went to the manufacturer's website to see what issues what the actual sequences they was looking for, but I
Oh,
couldn't find
nice.
them. Well, I could, but it was catered for, but it's directed to like respiratory samples,
Yeah.
which again have a much lower diversity of bacteria in them than say
Yes.
colon samples.
Yes.
So the
And actually,
chances of
the funny thing is like we can actually link to the deep dive that we did recently, right? Of like just like the fact that inside of the C. elegans genome, there's a homology site to something in small RNAs in bacteria, right? Like there's all sorts of
mm,
diversity of sequences to be found in large samples.
yeah,
Yeah, so like
and
if the doubt, right? So the doubt that you're trying to bring in here is like what about spiraless amplification? Right? Like
yeah,
you're using
spurious
this
amp.
kit, yeah, you're using this kit to go and like try to find like low abundances inside of a sample. Yeah, so what's happening there? Like there's that potential.
Yeah, but I think I've missed off the reason why I'm so skeptical. I think that because I think I need to point out that because I think what this paper is suggesting is that SARS-CoV-2 is somehow replicating inside of bacteria. Somehow the genome is being replicated inside of a bacteria,
Yes, yeah,
and
that's the hypothesis that they're bringing into this observation, right? Like the observation of the RNA finding, they're like, oh, maybe it's because it's replicating in a bacteria. And the two people that are in conversation in this paper, right, both sort of have the same idea behind it, right? Like the reviewer of this is also like is interested in this hypothesis and advancing this hypothesis
oh
specifically. Yeah.
yeah, I think that this needs some virologists to review it because
Yeah.
the people who are reviewing it don't necessarily have the experience, but I mean
No, I
there are
think
certain things
they say
like
that they are a virologist, though, the person who
oh
reviewed it.
really
Yeah, but
oh
but what it is like the person who's reviewing it is also after the same hypothesis, right? That
right.
SARS-CoV-2 is replicating somehow in a prokaryotic. It might have some sort of prokaryotic replication like
I
lifestyle
mean,
or
it's
something.
yeah, because yeah, it's one of those things. An extraordinary claim requires extraordinary evidence,
For
and this
sure.
paper
Yep.
hasn't really shown that.
It's just the beginning.
Yeah,
Yeah.
they do some like mass spectroscopy as well to try and find some peptide sequences. Again, that's
And
go.
they're also
I
trying to
haven't
go
been.
in the text of the paper, they say that they're doing like TEM of the samples, right? To like look and looking for associations specifically between viral like particles and bacteria. And also to try to exclude the idea of eukaryotic cells in their samples.
Yeah. Uh, I mean, what I'd be looking for here is actually like using another set of primers to check for. I mean, hope they created a cDNA library off of this somehow, and
Hmm.
that they're using their, uh, like, I mean, again, looking because it isn't
There's
like
a lot of
within
noise
outside
inside
the
of a bacterial
yeah
of a fecal sample. Like
yeah
there's a lot of DNA and RNA inside of that mixture. So then to go in with this kit, right, and amplify out certain things and say that they're increasing, like maybe there hasn't been enough done to like say that they've removed all sources of experimental error there.
yeah. For a paper like this, I'd be looking for like say a lot more genomics, so whole genome sequencing, or even just trying to like because we might be just looking for fragments as well, because seeing like amplifying out from those initial tests to see what they because it might be interesting if like bacteria are taking because we know bacteria can take up DNA. If they can take up RNA, perhaps what could be happening there, or like a CRISPR system perhaps could potentially is not outside the bounds of
I mean,
plausibility. Just
to me, I think that the best, like, if their hypothesis is like that it's actually making some sort of infective particle, I don't know if they're even going down that path, right? I think that,
I
like,
don't
based on the
know.
data that they've brought together, like, it could be like just like some piece of the RNA is somehow being replicated in a certain circumstances. But like, if they're going down the
And I
infective
can
viral
understand.
path, then they should just, or they should show the negative data if they're trying to collect it of just like doing the plaque assay.
Yeah,
Yeah,
I don't think there's like much evidence here for the hypothesis, and but I do feel like they because I can understand the logic behind it because there has been this like this contrast between viral load and viral titer, where
mm-hmm.
like the DNA of viruses do seem to of like SARS-CoV-2 does hang around a little bit longer. So I can see
Mm-hmm.
the logic behind looking down this path to see whether there's something maintaining that, and also like going back to some of the research we looked previously, where SARS has been spotted in the gut. Well, SARS-like particles have been spotted in the gut, perhaps.
Mm-hmm.
So I can understand the logic, but again,
But
the idea
in those
of
ones, we're thinking they're replicating in the gut cells, right? Like
yeah,
this paper is trying to say like not in the gut cells, but in the gut microbiota. I think that
yeah,
that's what stuck out to me, and I was like,
yeah,
this is such a strange claim.
yeah. That's where the bridge is too far, and that's why I mean, the thing is, like, if this is true, then it'd be really amazing. So I want them to actually present data to show whether it something to
So
explain
like
this as well.
yeah, and true meaning like true their hypothesis, right, is holding out. But the
Yeah,
data they've brought out to us, like in terms of like this replication, like okay, well that's showing us in a very limited way that you think that there's some sort of replication event. But like we want to see more lines of evidence for replication, I think. That would like really give some credence to this idea that it's yeah, making particles or something inside of the microbiome.
yeah. So yeah, I really would like to see a lot more like because I can understand when if you see that happening, then questions are raised because trying to
Yeah,
figure out why this is happening would be an important thing because it could be important thing for people who are just running SARS-CoV-2 tests anyway, just to make sure they don't make the go down the same rabbit hole or to understand what
sure.
could be happening to interfere with these tests.
Hmm.
Because again, there are people like who are doing sewage tests at the moment for SARS-CoV-2, so actually understand if this is something if this can happen, then that could be something that we need to pay attention to.
Yeah, it'd be a source of noise. But I think that like the
Yeah.
sewage tests, like they already have good models just like
Mm.
capturing, right? Because like they can correlate the sewage data to models of actual the cities infective rates, and then they have good. That's pretty strong. It's almost like it's a very
Yeah.
small contributor of noise, probably already accounted for, right, inside of some of those predictive models. I think that like any further like investigation on this road is like people have to believe or be interested in knowing, right, like whether or not there's some sort of event here happening in the microbiota.
Yeah, I guess I guess I tried my best for my devil's advocate work on this, but
No, that's
uh
good. No, I mean that's why I
yeah.
picked it out because I thought it was interesting. You also get to see, right, like the way that this paper gets published is you can see who the reviewer comments and what's being done to address them. And as I said, like it's interesting to see that like one of the reviewers here also is interested in the same hypothesis. So there's
Yeah,
definitely
that's
that. Yeah.
it does shine a light on like how research works, I think, and
Mm-hmm.
how peer
Mm-hmm.
review works. Because sometimes the only people sometimes people who review your work are people who like believe in the hypothesis. Sometimes
Yep.
it's the opposite. So it's kind of chaos. So which is why I think it's always important to like instill like a critical eye on anybody who has to look at these papers for their work,
Totally.
because yeah, because you can't necessarily rely on the review process to pick out like every paper that's bad, because
Yeah, yeah,
reviewers are still people with their
yeah.
own biases and their own like kind of thoughts.
Hmm.
So I mean, I think like that's so
Yeah.
like
And
I
so
think like
a lot of like doing good work in terms of pushing some science forward is also choosing what critical things you want to be able to replicate in your own
yeah,
hands, and then extending that one step further, right? Like using, as we were saying, like another line of evidence to just give yourself that confidence, right? That you're going down. Yeah, you're making the right assumptions as you go to the next question to ask.
yeah. That's why sometimes papers can feel like very much like, oh, I've heard this before, because a lot of researchers spend a lot of time trying to prove things in different ways
Yes.
until like, and usually like I get convinced by a paper's argument about halfway through the paper before they lay even more data on me.
Yes, yeah. And then the
Because
last part is like,
yeah,
oh yeah, like that just it just clicks in and you're like, wow, like I'm so glad that they did that in this other way because like I wasn't even too concerned about that corner case, but they removed the doubt by doing this other piece of this other experimental approach.
yeah, because papers are read by a multitude of people with different experiences. So
Hmm.
so it depends on like, so if you have a diverse set of reviewers who are reviewing a paper, you're going to get all sorts of adaptations based on those people's experiences and what they bring to the table.
Yes, yes. Yeah, so I chose this because the the title really got me. The fact that we could see the reviews I thought was really interesting. And actually this like kind of linked up to, do you remember like I was talking about like the preterm babies microbiota transplanted in germ-free mice and seeing those outcomes,
Yes,
and just like
I
how
remember.
early, right, like how that like early type research is done, right, where it's like, yeah, it's like just an interesting thing, and like there still has to be follow-up to understand like what that relevance is in any circumstance. Here it's like even more tentative because like
Oh,
like all they have is the amplification. Everything sort of hinges on that.
yeah, because I feel like this paper feels a lot on the same level as those preprints we were talking earlier, but this one's
Hmm.
got to add 2 reviewers, so but I think it's different for different fields. So it's hard to like compare across different places. So
Yeah.
um,
Okay.
but yeah,
That's all I want to
okay,
say.
yep. And now we're going away from
um,
SARS-CoV-2 entirely to
yep.
uh, E.
This
coli
is,
actually.
yeah, anti-E. coli. And this is like, okay, I feel like maybe this week I chose papers like based on like just how they're interesting about showing like an aspect of science research. And this one is about so in E. coli you can have like if you get dysentery from E. coli, or like if you get food poisoning, it's often through a Shiga toxin that's being made.
Mm,
So like basically the pathogenicity of enterohemorrhagic E. coli is based on a toxin that's expressed from a phage element.
yeah. So like phages are viruses that infect bacteria,
Hmm.
and yeah, and it's interesting because like there is a diversity of different E. coli. Some are actually but really benign,
Yeah,
but
they do
then
nothing.
you got like enterohemorrhagic, which is I think Latin for bloody guts.
Yep.
Yeah, because that's what it gives you. I think it's
Yeah.
evolved to live in cows because cows have much stronger guts. So in order to like isolate their place, they have these toxins that really like help it like nail down. And then when it goes human, they come with the humans with the same energy, and it causes
We're not
us
able
to.
to, yeah, we can't deal with that, and it ends up in like it's quite a dangerous thing to have actually.
Yeah,
And so like this is like a major foodborne illness, as you were suggesting. They live in cows mostly, and when agricultural runoff gets into liquid, right, and
mm,
um, yeah, it gets into
yeah,
water supplies or stuff like that, or food supplies. That's when
yeah,
this has really bad effects on the human population. So again, that toxin is being made on a phage-encoded element, and in this paper, there's already some knowledge known that small molecules made by E. coli actually stimulate the production of that toxin, and so they're following up on sort of all of these different small molecules, collicins actually, which
yes,
when made in very high concentrations are a type of antibiotic that we use and are investigating in terms of synergistic effects with other antibiotics for
yeah,
therapeutic use. But all antibiotics have some basis in nature. They're not just like, I mean, they're basically to kill for the bacteria that use them to kill other bacteria that they don't want. But then there's like evolution. It's like, well, actually, if you're getting killed, then maybe you need help in surviving in that niche, and so maybe this toxin's going to help you. So there's already been a link between the production of these collicins and then the production of this toxin. And in this paper, they look at like a tiny version of the collicin, a microcollicin, that or sorry, a microcin, microcollicin, a
mm,
microcin, same sort of idea, small molecule,
yeah.
potential antibiotic activity, and they're following up whether or not it induces the effect of stimulating um, this toxin production.
Yeah, so I think they do a bunch of different like
They make the
experiments
mutants,
on it.
right? This is a
I think
very, yeah. To me, this is
I
like
think
this is the type of science that I have always that I really enjoy, like in terms of like the way that it shows stuff. It's just it's curious about how this thing works, so it removes it and it does an assay, right, where they have it removed from the strains, whether or not, yeah, the it can be turned on, the toxin can be turned on in the presence or absence of it.
yeah, this is interesting because we often think about microcins or like collicins as like antibiotics. They kill other bacteria, but here it almost seems like it's being used as a signal for other bacteria. So that at
Mm.
low doses, it causes it like kind of makes E. coli go through this stress response to produce the Shigella toxin.
Yeah.
So
Well, I
that
mean, I think the stress response is potentially right. Like, it elevates like I don't know, like group stress or something like that. But
yeah.
like the Shigella toxin, it itself might be a mobile element, right? That's moving around. So we get into that realm of stuff that we've talked before. Is like, what is the purpose of having this toxin, right, on board E. coli? Um,
Yeah, and they take it into mice to look at like how that affects like colonization, and
mhm.
yeah, uh, I mean, yeah, it's interesting. They find a receptor for it, and yeah,
Yes. Yeah, they find it's the
it's in,
same receptor as the regular collicid, right? Because this is like this microcin is like is a class of molecule that I guess doesn't have a lot of data to it. So in some ways, what we're seeing in this paper is extending some of the things that we think about collicins to this microcin as well.
yeah, and actually this ties into like a review I read earlier this week about what how we need to rethink how antibiotics work in nature because maybe
Mm.
they have other functions other than killing other bacteria, and
Yeah.
they're part of
Yeah.
like resistant systems, and that we don't fully understand how they worked in nature when we
Hmm.
first used them, and now because of that resistance evolves very quickly because it was kind of always there as part of
Hmm.
like bacterial communication. So
Yeah, because it's like
I'm yeah.
they have networks to respond to these molecules already, right? Genetic networks, right, that have been like sort of shaped through evolution already to respond to them. So like when we increase the concentration of them for our own therapeutic purposes, maybe those networks actually have a lot of capacity still in them to flex around and say, okay, actually no, we don't need to respond or die off to this thing anymore. We just will ignore this object.
Yeah, it's interesting. I never really thought about things like that, and I feel like I paraphrased someone's review really badly. But the great thing about that is I haven't cited either, so they can't catch me. So
And actually, wait, that just reminds me of another thing that they found in this paper is the genetic module that makes this microcin also has like the antitoxin elements associated with it. So like, right,
yeah.
like the microcin does have an antibiotic, an antibacterial effect, but the same operon that makes it also makes some other molecules, some other proteins that neutralize it. So like this is
That
also a
yeah.
theme that we'll see. We haven't talked about it too much, but like this is a theme in bacteria where they have like
Oh,
toxin antitoxin systems. They make the thing that then they also neutralize. So then they can give the toxic thing into the environment or into other bacteria, and they don't have to worry about killing themselves with that molecule.
I mean, that's a very something because I found like you see that in phages a lot as well. I think where
Mm-hmm.
you get phages that have like some kind of thing that kills off other bacteria, and then cause I think we did like we did deep dive into one of those papers where we had like two phages fighting each other using bacteria to do it. It was
Yeah.
a wild paper. My favorite ones we covered. Got very low views, unfortunately.
Well, then we should wait. We
But
should. What's the title of that one? We should just point people to it. That was
oh, I
Guns for
think
Hire.
viruses that fight each other.
Yeah,
I
Guns
think
for Hire.
yeah. The like
Okay.
my favorite story in microbiology is completely useless for like understanding health, but it's just a cool story. Um,
Okay, let's move
uh,
on to the next paper that we have in the docket.
yes, a host-directed macrocyclic peptide therapeutic for MDR gram-negative
Mm,
bacterial infections. So
yeah,
this, if I'm not, yeah,
go ahead.
uh, a, an antibiotic from derived from apes apparently. Um,
Yeah, and not like a direct the macrocyclic peptide. It's like not directly affecting the bacteria itself. It's producing it's priming the immune system to act in a certain way against the bacteria.
Yeah, well, this is interesting because when you put it in a petri dish with bacteria, it kills off the bacteria. But when you add like aspects of the immune system and the bacteria, it doesn't kill the bacteria because but it preferentially binds to the immune system as well.
Hmm.
So and then it seems, but even then, it seems to have an antibiotic effect when you use it in a model of sepsis.
Yes, in a
So
model, yeah. I guess like the question, it's kind of to me, it's a very strange molecule, right? Like, what is it doing exactly? Is it stimulating the immune system? Is it having a direct effect on the bacteria itself? I don't think they really separate that
so
necessarily in their model.
I don't know. But I think like
Yeah.
because they're like a type of defensin, so humans produce these antibiotic like molecules as well. So I think,
Yes.
but here they use theta defensins, which are only found in apes, because I think there is this worry that if you create one that's based on humans, antibody when bacteria become resistant to it, they also become resistant to the native version we all produce, so they'll become more
Mm.
virulent. So that's
Mm.
why people are looking for other species for defensins. But yeah,
Oh,
I
that's
think
interesting.
because I think it also like harks back to like evolutionary, like an ancient form of like antibiotic resistance, sorry, bacterial resistance that we had, where like back when we were like kind of jellyfish, we didn't have like a fully amazing immune system, we'd produce like antibiotics like this. Um,
Yeah, just like but I think also, but then these antibiotics have signaling functions in our immune system,
yeah,
right? Like that's the other thing. Like we're moving a potential signaling molecule from one context into our context and seeing that actually, yeah, it could have some therapeutic abilities in us as well. Well,
yeah,
I
I
guess
think
in mice, right? Because that's what they're doing. Oh, and they do it in with cell culture as well with
yeah,
THP cells.
yes,
Well, not
they
THP,
do.
but macrophages of some sort.
Yeah, so so these things evolved ancient, and now they're part of the signaling system because our immune system is much more sophisticated now. Uh,
Mm-hmm. Mm-hmm.
yeah. Um,
Yeah. I just thought an interesting class. So yeah, I picked it as because an interesting class of antibiotic that I hadn't really
yeah.
thought about.
Let's see. The next one is a nanocompartment system contributes to defense against oxidative stress in Mycobacterium tuberculosis.
Hmm. Yeah, so this is nanocompartments. Uh, so bacteria also have organelles, or it's coming.
Yeah.
People are beginning to understand that there are organelle-like objects inside of bacteria. they're not lipid separated, right? They don't have a lipid bilayer that separates them from the rest of the cytoplasm. They have like some sort of like proteinaceous coat. So
Yeah.
essentially, they're like capsids. Actually, I find that
Yeah.
they're very similar to capsids inside of cells, and then they can isolate specific enzymes that do different things. Maybe they need a specific environment, right, to work. That's what these nanocompartments are.
Yeah.
And in this particular paper, they show a function of one.
Yeah, so they find like we spotted nanocompartments in like other bacteria, and what they found was a similar gene in tuberculosis, and then so they knocked that gene out to see what would happen, and they also did some imaging to show maybe these nanocompartments inside tuberculosis. Um,
Mm-hmm.
and yeah, because one of the things like immune systems do when they find cells is they do this oxidative burst. They produce a lot of like oxygen like free radicals that are very
Mm-hmm.
damaging to the bacteria and to cells generally and to life. But
Yeah.
tuberculosis has peroxidases that can break down these reactive oxygen species and
Yeah.
detoxify them.
And
And the
it's really
theory is that
important.
these
It's really important to the tuberculosis life cycle because, like, tuberculosis is recruiting a bunch of macrophages to it in these big structures,
yeah.
and so being able to detoxify that scenario and thrive basically inside of this dense. I think they even call it like cheese-like, right? Like
Yeah,
caseous,
granule, yeah, caseinaceous
caseinaceous
granulomas, where they,
granule. Yeah.
yeah, they form these like kind of like separate like I cankers
Pus-filled.
inside your lung,
Yeah,
I guess,
yeah, yeah.
like
Where they
these
c-
like
where they're isolated from the body as a whole, but they're also sort of feeding off the nutrients of all the dead cells in that environment.
yeah, and they live inside immune cells, the very cells that are designed to destroy them. So they have
Yeah.
very sophisticated ways of trying to combat those immune cells, and this is one of them. And
Yes,
yeah,
one of them. Yeah, so then they go in and they find the genes that are responsible for making those particular nano compartments that make the peroxidase. They knock them out, and they find that if they remove them, then yeah, the bacteria get eaten up easier, or they aren't as tolerant to oxidative stress.
yeah, they don't survive as well. And this
Yeah.
like is the kind of research that always makes me kind of really interested in terms of because we found like an aspect of bacterial metabolism that's very different from humans, which means that we can drug target it potentially. So
Yes.
and use that could be a good target for antibiotics potentially.
Yeah, like the whole yeah, the whole process of these nanocompartments being assembled, there's probably many things that have to go right. And if you can find a small molecule that gums up the works, then yeah, you've got a great, yeah.
Yeah. I feel like once they get the protein structure of this out, that there'll be lots of people with their computer models like throwing random like molecules at it to see which one sticks, and then going ahead and doing a panel, and then maybe we'll get a new tuberculosis drug. Um,
Yes. Which is so important because I think multi-drug resistant tuberculosis is like a big problem.
yeah. So yeah.
Okay.
Next one is
And our
uh,
final paper of the day, colonies of the
is
marine cyanobacterium trichodesmium optimize dust utilization by selective collection and retention of the nutrient-rich particles. This is like looking at dust and small molecules under a microscope all day and characterizing the behavior that they're seeing. There's this cyanobacterium that forms little puffballs in the marine environment. And these researchers collect a whole bunch of them and treat them either with dust particles enriched for a certain type of certain nutrients, either phosphorus or iron, and they just look at the how many of these particles get associated with these collected bacterial colonies, and they find a pattern that they really want to collect dust particles that have the nutrients that they need.
yeah. It's really interesting because I think like the Sahara plays a massive effect in terms of ecology because while it seems like there's not much life there, when dust storms blow out into the sea, they can colonize the ocean, and and bacteria like this can suck them up. And this is kind of
Yeah.
trying to figure out how smart those bacteria are. Like, do they just suck up things every single thing that looks like a dust particle, or do they check whether it's got like the nutrients they need? So does it have the
Yeah.
phosphate? Does it have iron? And if it
Yep.
doesn't, then apparently they just go, no, we don't want that. And
Yeah.
this,
Yeah,
I like,
this paper
yeah.
is very like it's almost like a like a animal behavior, right? Like you went into the forest and you looked at a bunch of animals and you saw them prefer certain things over others. This is just like the micro version of that. They like went into seawater, they picked up puffballs, they gave them the choices of collecting like either pieces of dust and they just counted, right? Like what's the frequency of these different behaviors?
Oh yeah, don't get me wrong. I'm very jealous of these researchers because they went out in a motorboat and collected stuff like over a series of days. Like here was me in a lab with like vitamin D deficiency and just like not getting any sunlight, and then they're out in a motorboat doing like cool marine biology.
Yeah. Well, but then they had to go back into the lab and they had to look at each little puff that they had, right? Like over a
True,
period of
yeah.
time. So don't worry, they spent spent a lot of time in the dark lab as well.
Yeah,
And a
and they
stereo
also collected up
microscope.
like normal dust particles. So they collected them from like their roof and window shades, apparently, like on flat, clean surfaces. And then they tested them, and they found that no matter what, the bacteria preferred those over everything because I imagine that those had a combination of different nutrients. Um, and another thing is like this is one of those instances where you get single-celled organisms that behave like multicellular organisms. They're a colony that acts together, and they like draw in these
Yes.
particles to their center.
Yes. Yeah, I think that's so cool. I mean, they show it here. They have like these are like the diagrams and essentially what they were looking at, right? Is like these little strands. And they also say that actually if you go to oh, I don't even have the figure numbers here, but it's like oh yeah, this figure. Yeah, those are actual images of them, I believe.
Yeah.
Yeah. And so you can see like they're just like these very spindly things and over time, right, the particles get centered into these colonies. So they
Mm.
get stuck on the periphery, but like the longer they observe these little puffs, like those small nutrient filled yeah, ostensibly nutrient filled dust particles, they move to the center. So there's definitely some sort of division of labor and like yeah, sort of multicellular cooperation going on here.
Yeah, I think that this is a cool paper and definitely worth a
Oh,
look.
and, and if you go down a little bit more, I just wanted to point out they also found two like more yeah this one two morphotypes, right?
Yeah.
That they saw that like based on how dense the sort of fibers were, they actually had different behaviors. That the ones that were thinner are essentially in like a collection phase, like they really need to collect things, but the ones that are denser, they're kind of like it's fine. They can just like have
The
some stuff stick on the outside.
right, so they become like less active when the bigger they
Hmm.
are. So yeah, like
Yes. Yeah.
the fat
They're
colonies
slowing
behave
down.
differently.
Hmm.
I mean, part of me just thinks of Halo and oh my god, they're going to form a grave mind if we let them grow too big. But
That's probably true for a lot of bacteria. That's my hope, actually, right?
why is that your hope? Have you not seen Halo? Oh god, horrifying.
Oh no, I don't. I guess I don't know what happens in that particular video game, but
Oh,
like, to me,
sorry.
it's like these are like the advocates for the environment, right? Like, if they could come together and sit on some of our political boards, then we'd have a totally different perspective on the world.
I mean, I feel like a few people on our political boards are already viruses, but that's enough politics for today.
All right, so those are all the papers that we have for you today. Um, Join us next week where we're going to continue to cover more microbiology news just like this.
And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have questions or corrections, please let us know in the comments.
Yes, I totally agree. You can reach out to us over Twitter at microtwjc. We both believe that peer review is a process and that we're all participating in it together. So if think you have something to add or found something unclear, just let us know.
Yeah, it's been a pleasure chatting with you, Danny.
Same here, Fuzz.
All right, bye.